The Epidermis PhysioLab platform is a unique, powerful, and flexible research tool. It is a mathematically explicit dynamic model of bioprocesses that take place in human epidermis, leading to formation of barrier function. As such, it is a powerful tool for studying the interactions that take place in viable epidermis and stratum corneum, which affect epidermal quality. It allows for studying these interactions in a matter of minutes or hours, as opposed to weeks or months. The platform was specifically designed to support a variety of uses in skin bioscience research and development, and support scientific investigations and compound development. Using this platform, researchers can quickly formulate and run so-called in silico experiments, that is, virtual experiments running in the computer that explore many different what-if scenarios. For example, what if we could hit target X? What would be the clinical effect? Or, what if compound Y's mechanism of action hits this pathway? Would that be consistent with the evidence we've seen so far? The Epidermis PhysioLab is flexible in the sense that it can be customized to account for new or additional data. Both normal skin and cosmetic dry skin are represented in the platform. Furthermore, because interpatient variability is a big concern in this field, the platform gives researchers the ability to explore many different dry skin subtypes. This means that novel treatments can be tested against a wide variety of so-called virtual consumers, representing different subtypes of consumers. The platform includes a number of treatments and perturbations, including tape stripping, application of acetone, glycerol treatment, occlusion, and application of sodium lauryl sulfate. These different virtual experiments are included in the model both for validation purposes, i.e., ensuring that predicted responses match reported clinical responses from the literature, and to serve as templates for additional treatments that the user of the platform may want to explore. Because the ultimate goal of skin treatments is to improve skin condition and appearance, the platform reports a number of cosmetic endpoints of interest, including barrier function and transepidermal water loss, hydration and natural moisturizing factors, pH, epidermal thickness, desquamation rate and quality, visual assessment of dryness, and elasticity. This allows researchers using the platform to immediately assess the impact of a novel treatment on clinical endpoints that they care about. The platform captures keratinocyte and corneocyte biology through the proliferation, differentiation, and cornification processes. Cells are tracked as they move through the different strata, from stratum basal through spinosum, granulosum, the transition zone, and finally to the inner and outer stratum corneum. Biological processes that take place during the keratinocyte life cycle are represented in appropriate layers of the epidermis. Along the way, various regulators come into play to govern these biological processes. These regulators include calcium, pH, water activity, and various cytokines and growth factors. When skin conditions are perturbed, for example as a result of treatments or experimental perturbations, these regulators dynamically restore the skin to normal functioning. This slide elaborates on the process used for validating the platform. To validate that the platform's response to treatment and perturbations is appropriate, a vast amount of literature was reviewed. For each type of experiment, the most common versions of the protocol were selected and simulated in the platform. Measurements were taken at the same times as in the literature experiments, and as many of the endpoints were compared as possible, given the data available. The platform reproduces the homeostatic state, as well as appropriate responses to tape stripping with removal of most of the stratum corneum, application of 2% SLS patches for either 24 or 48 hours with 14 days of follow-up, application of 15% glycerol twice a day for 10 days, application of petroleum for 60 minutes as an example of an occlusive, and application of a solvent, namely acetone, looking at a single application. Specific literature references used to compare to are cited on this slide. The statement that the platform is validated means that it is capable of reproducing appropriate responses to all of these virtual experiments without any adjustments of model architecture or parameter values between experiments. In addition, the model captures the appropriate time profiles of these responses. The validated model can then confidently be used for predictive biosimulation research.